Great.
Thank you for the introduction, and thank you for the opportunity to present our work here. I think I'm going to demonstrate the breadth of the research supported by the Duke CFAR by changing the topic into a molecular biology research. So we're interested in the Colin lab in epitranscriptomic RNA modifications and how that affects viral replication, and how that might provide a potentially new avenue for antiviral development. All right, so I'm sure most of you are familiar with the central dogma of gene expression, where the information encoded in the DNA is transferred over to RNA and to protein, which confers biological functions. Now, this pathway is subject to further regulations, such as epigenetic modifications that could modify how the information encoded within DNA is interpreted. Until very recent years, we have started to realize the importance of further regulations on the RNA level, which is analogous to epigenetic modifications. There are also small chemical modifications on RNA, termed epitranscriptomic modifications. And we are interested in how these RNA modifications can regulate viral replication. So what exactly is epitranscriptomic RNA modifications? Examples include methylation, the addition of methylations and acytylations on RNA bases and the backbone as well. And they provide an additional layer of information on top of the mRNA sequence. They may regulate RNA and protein interactions by either the addition or the removal of interaction sites. And this may manifest in changes in any step of RNA metabolism, ranging from splicing, RNA export, translation, and RNA decay. So RNA modifications has been found on viral transcripts since the 1970s, starting with the most abundant RNA modification, M6A, which is a methylation on adenine on RNA. However, up until the 1980s, we don't really know what these modifications do. In fact, back in 1987, a paper suggested that M6A doesn't really do much in the context of Rous sarcoma virus. It isn't until the recent years where we see a recent renaissance in viral epitranscriptomic RNA studies. This started with M6A studies on HIV-1 and flaviviruses, which include 2 studies that are from here at Duke. So over the years, we have found specifically the most abundant RNA modification, M6A, on the transcripts of a wide variety of viruses, ranging from DNA viruses, RNA viruses, to retroviruses. And what's interesting to us is that RNA modification seems to mostly enhance the replication of most of the viruses reported so far. And also, in collaboration with Chris Hawley's lab, we have found that the HIV-1 virion RNA is highly enriched in several RNA modifications, including the methylations shown here, up to a 20-fold enrichment of 2' methylations on the viral RNA as compared to the cellular RNA. And so we think that viruses may have evolved to incorporate RNA modifications that are beneficial to the virus life cycle. And that since RNA modifications affect a wide variety of viruses, this may represent new targets for broad spectrum antivirals. So a bit history on what the Colin lab has done in this regard. We have studied several RNA modifications in the context of HIV-1 replication. First of all, we have studied the methylation of adenine, or M6A, on HIV, and we have found that the recognition of M6A by the reader protein YTHDF2 results in increased HIV RNA expression levels. We have also expanded into studies of another methylation on cytidine, namely M5C, and we have found that this increases mRNA translation from HIV-1 genes. And our most recent topic involves an acetylation on cytidine AC4C, which is the main topic of this talk. So AC4C, or N4-acetylcytidine, is the addition of an acetyl group onto cytidine on RNA. This is previously found on tRNAs and the 18S ribosomal RNA. And we also know from prior literature that the cellular enzyme N-acetyltransferase 10, or NAT10, is the enzyme responsible for catalyzing the acetylation of cytidine, forming AC4C. Back in 2018, Shalini-Oberdorfer's lab at the NIH has also found that NAT10 can deposit AC4C on mammalian cellular mRNAs, and that AC4C acetylation on mRNAs can increase, with encoding sequences, can increase the efficiency of translation and increase mRNA stability. So we're interested in whether this could be involved in the regulation of HIV-1 replication. So to start off, we utilized an AC4C targeting antibody that's recently available, and we used this to pull down AC4C acylated RNA from either intracellular RNA from mock-infected cells, from infected cells, or from viral particle encapsulated virion genomic RNAs from 2 different T cell lines. And as you can see, as highlighted in the figure here, we have identified 11 distinct sites with AC4C present that are consistent across the intracellular viral RNA, the viral particle RNA, but not present in the mock-infected intracellular RNA. As previous literature suggests that NAT10 is the enzyme responsible for the acetylation of RNA, we wanted to test whether if our AC4C identified AC4C sites are NAT10 dependent. To do that, we first utilized CRISPR-Cas to deplete T cell lines of NAT10, and as you can see here, most of the AC4C sites are either diminished or gone in cells depleted of NAT10. And we can also cross-validate this by doing a PAR-CLIP, which is a protein-RNA interaction assay, where we can find NAT10 binding sites on the viral RNA. And here we have found that the NAT10 enzyme indeed co-localizes with most of the AC4C sites that we have found. So this suggests that the AC4C acetylation on viral RNA is dependent on NAT10. Next, we're interested in how this affects HIV-1 replication. To test to understand what's the most important step in the HIV life cycle that's regulated by NAT10, we did a single round infection by adding a reverse transcription inhibitor called nevirapine at 15 hours post-infection, which is sufficient for only 1 round of infection. So we tested 3 different independent single cell clones of the delta NAT10 cell lines of the CEM T cell line. And indeed, we see a drop in viral GAG protein expression in all 3 independent cell lines. We also see a similar drop, we also see a similar drop in RNA levels, where we see a 70% drop in HIV-1 RNA expression from the delta NAT10 cell lines. And we tested multiple steps within the RNA metabolism pathway, and we have narrowed down the main impacted step in RNA metabolism is RNA stability, where when we isolated newly transcribed nascent RNA in infected cells, we have found that the viral RNA is decaying faster in the delta NAT10 cell lines as opposed to the control cell lines. So this suggests that NAT10 deposition of AC4C can regulate the RNA, can enhance the RNA stability of HIV-1 transcripts. However, most of our prior studies focused on NAT10, which can also deposit AC4C on other non-viral RNAs. So to be more specific, we wanted to specifically remove AC4C from only viral transcripts. To do that, we targeted a couple AC4C sites that we have mapped namely peaks number 4 through 8. And we identified any Cs within the mapped AC4C site region that we can mutate without changing the amino acid sequence. And as expected, from the wild type or the AC4C depleted mutant virus, we see that the mutant virus showed a 60% drop in viral production levels. If this phenotype is indeed dependent on NAT10 deposition of AC4C, we would expect this phenotype to be diminished or gone in delta NAT10 cell lines. So if we normalize the viral titers by the P24 levels, viral particle levels, and then infect either control or delta NAT10 cell lines, we would expect that the difference between wild type and mutant to be more pronounced than in the delta NAT10 cell lines. And indeed, that is what we have seen in this Western blot here, where we see HIV-1 P24 protein expressed at a lower level from the mutant virus in the control cell lines as compared to the wild type virus. Whereas the difference between mutant and wild type virus is less pronounced in the delta NAT10 cell lines. And we can calculate the wild type to mutant ratio in this figure here, and we see that it is a smaller ratio in the delta NAT10 cell lines, suggesting that this phenotype is dependent on NAT10 deposition on AC4C. Lastly, from prior research, we know that there are a couple of small molecule drugs, including 3-deoxyadenosine or DAA, that could inhibit the methylation of RNA, the M6A methylation of RNA, and this is antiviral due to the fact that it could inhibit M6A enhancement of the replication of several viruses, including HIV-1 and flu. So analogous to this, we wanted to test a small molecule drug that could inhibit NAT10 acetyltransferase activity. This is called Remodelin. And indeed, at concentrations that we tested that had minimal effect on cell replication, we see up to a 70% drop in viral RNA expression levels. If this is indeed inhibiting NAT10, we would expect the phenotype to be diminished in the delta NAT10 cell lines. And indeed, we see that the RNA levels are consistently down in the delta NAT10 cell lines. So in conclusion, we have found that the HIV-1 transcripts are AC4C acylated, and this is dependent on NAT10. And we have found that NAT10 deposition of AC4C can prevent the premature degradation of HIV-1 viral RNA. And that we can also phenocopy this by using a small molecule NAT10 inhibitor called Remodelin. And this suggests that that further research into viral epitranscriptomics could lead to new targets for antiviral drug development. So with that, I would like to thank all the past and present lab mates that have helped with my studies, our collaborators, and our fearless leader, Brian Cullen, and funding support from both the Duke Center for Virology and the pilot grant from Duke CFAR. So with that, I would like to thank you for your attention, and I will be happy to take any questions.
Great, thank you, Kevin. Congratulations on the really beautiful work this year and your publications.
Thank you.
Can you comment on how this might be altered with the activation state of T cells? I know you had to use a T cell line,
Right.
but expression levels for NAT10 and how that's modulated with activated CD4 T cells.
We haven't specifically tested for that, but I can tell you that the AC4C mapping results from activated PBMC isolated CD4 positive T cells looks pretty much similar to what we have seen from cell lines.
Great, thank you.
Thank you.
Did anyone else have any questions they wanted to ask? Okay, nicely done. And then just one final one. This also relates to cell types. Are you thinking about looking at this in other cell types, macrophages, for example?
Yes, that would be interesting. And yes, that would definitely warrant further looking into.
Okay. And what do you see as a path forward? You know, your last comment here is about the antiviral effects
Hmm.
and how you can modulate that. Do you what would you foresee as potentially the next steps to see if you can utilize this information to have a therapeutic intervention?
So we're basic virologists, so we're more interested in the biology side. However, so what I'm interested currently is two main directions. First of all, whether if the addition of this acetyl group could either inhibit or promote certain RNA protein interactions that could affect the outcome of viral replication. And furthermore, I'm also interested in how metabolism could affect this acetylation, since the donor of this acetyl group is acetyl-CoA, and that could provide implications on, for example, HIV-1 replication in diabetes patients.
That's interesting. Great. Thank you very much for your talk. Well
Okay.
done.